mostly we're going to talk about B cells. Oh, that's what it is. I was wondering if somebody forgot it here.
Is it called smelt?
Smelt is the fish that.
Yeah, smells like cucumber.
Yeah.
Does it say? Have a European smell.
Is this going to
That
be
smells
a
like cucumber.
question on the test?
You can bet it's going to be a question on the test.
Smelt.
You didn't like my Montana question last time?
I did. I loved it because I got it right.
Let's get together before the exam and talk about what you would like to see. And then
Right. Extra credit.
So you'll hear mostly about T cells, B cells, and how they cooperate to make specific immunity happen. And then if you have time, we can go through a few slides on immunization, which is interesting, what they do, and there's some interesting biotechnology happening with monoclonal antibodies, things like that. But so this whole chapter is about specific immunity. And I cut out of quite a few things to make it shorter and more dense, but less polluted by things you don't have to know. So the system, as I said, the third line is acquired, meaning you're not born with it. You have to be exposed to microorganisms to actually develop immunity that or develop the cells, the B and the T lymphocytes that you that then can go on to give you immunity for certain diseases, for certain infections. And immunocompetence is really what you build after birth. So that's what the main term for it was. Your immune system becomes competent. So you're born with an incompetent immune system. That's another way to look at it. And a big term you always hear in relation to immunity and immune system is antigen. So antigen is everything that stimulates a response. So if you have pollen, for example, from a plant, that's what this picture is showing. Or if you have blood from another person, or if you have microbes, viruses, bacteria, all kinds of other organisms. Antigen just means your body recognizes those and responds with a T and a B cell reaction. So they have to be exposed, but a lot of things, even if you have an autoimmune disease, for example, then your own body presents antigens to your T and B cells. So you're basically, instead of recognizing the proteins or the structures on the cells as your own, they're recognized as foreign and treated that way by the T and the B cells. And so it's a big group. Anything that can do that. So antigen just means, and it's foreign. The T and the B cells respond, and a good way to remember it too is you have antibodies, and they react to antigens. So they're the two opposites. And we get to antibodies soon. The most important thing for the specific immunity is the specificity. Makes sense. It produces antibodies, which we just mentioned, maybe for the first time, against antigen, and they were produced in response to an antigen. So they're not just there. So it's specific to an antigen. And antibodies are produced by B cells, but I get to how they do that too. And then the memory is another one. So it's specific, and there's a memory involved. There's a way of memorizing those antigens and being able to react to them the next time they enter your body. So in general, what you need the specific immune system for, or how it works is your immune system develops and differentiates. So the B and the T cells are going through a process in the thymus and in the bone marrow, and you can already make that distinction. So if you have a B cell, they all originate in the bone marrow, but then the differentiation and the maturation, so to make the B cells, what actually you'll find in the body happens in the bone marrow, so they don't really leave. So let's call it bone. And then the T cells are going through that process in the thymus. So just T, C, E, D, just so you have an idea of where that happens. Another thing that the specific immune responses, or a better way to say that is the processing of antigens and what lymphocytes do with those antigens is part of the specific response. So the T and the B lymphocytes react to specific antigens, and then these B and T cells actually work together, so they cooperate during antigen presentation. So it's, as I said before, some cells are able to not only kill the microorganism but extract a piece that they think is important and then present it to specialized cells to say, look out for this. And then these cells say, okay, we're going to produce a bunch of soldiers, if you will, that are all looking for the same antigen. So that presentation is really important, so that you have a lot of cells looking for those and producing antibodies against this antigen. And that's part of what this is, the B lymphocytes and the production and action of antibodies. So these antibodies that are working against or binding these antigens are produced in B cells, and then you also have T lymphocyte responses. And as I said, these two work together. So the B and the T cells are both active immune cells that respond to specific antigens. And then back to the receptors. If you have, we're going to talk a little bit about receptors. So far, you've heard about these receptors that are able to recognize pathogen-specific or pathogen-associated molecules or molecular patterns. So there's these regular receptors that you find, but these specific cells have receptors that are, or they build receptors that are more specific to antigens. So what the receptors of some immune cells have to do is they have to be able to recognize and attach to foreign molecules, or to non-self is another way to say it. They have to also recognize the cells, so they don't, these B cells, T cells do not attach or do not attempt to destroy your own cells. They need to receive, and so another option or another part of it is they need to receive and transmit chemical messages, so chemotaxis can happen. And it's just this is just a summary of what the membrane receptors should do, and they should aid in cellular development, meaning when the cells receive certain signals, they can develop specific cells then maybe producing antibodies, et cetera, et cetera. So mainly, I would say for receptors, you should know that they're really important in recognizing non-self or foreign molecules, and then also initiate a response by, for example, sending out interferons or other chemicals that attract more white blood cells. So I'll show you two, and this one is really important. So the main purpose or the main message is those cell receptors and they sometimes call them markers. So they kind of mark what cell you're talking about. They give specificity and identity to a cell. So if I, for example, and we get to these, if every single cell except your red blood cells, every single cell in your body, doesn't matter if it's in the brain, if it's your liver, they all have these MHC1, and that's this one. So they all have the same receptor. So yours are different from mine, but they all are the same on your cells, and they call it MHC1 or major histocompatibility complex 1. And the complex really is the name for this major complex relates to the genes, but all you need to know is that this receptor, is obviously has a genetic background. You have the genes for it in your DNA, and it produces this receptor MHC1, and you have it in every single cell. So the main important thing about this is you can remember it by thinking about what your immune system does with foreign material, like say with an organ that's transplanted, and it's not compatible. So histo means tissue. Usually histology is the study of tissue, and it's compatible. So is your tissue compatible? Your tissue is not compatible with mine because you have a different MHC1 receptor on your cells. And my cells, my white blood cells, are going to look at this and say, that's wrong, you have to attack it. And so I'm constantly trying to get rid of it. So that's the way even though we're one species, we're not always compatible. Another one would be another additional receptor or structure would be our blood groups and how we're not compatible for those. How we have specific groups that we have less, obviously, because this is different in every single person. But one other thing is, what I was going to say is, if you then go in a family, my parents have different MHC1 molecules or receptors than I do, but they're not as different as some person on the road. So if, that's why you would then go and say, if there is a way of receiving organs, for example, or getting a transplant, then you usually look closer in your family, because the rejection of this is not as strong as from somebody that you have no relationship with. You still have, okay. So there's that, and I don't know. It's really hard to. I don't know what exactly those differences are in terms of. You would say, well, you either have a certain structure of a receptor or you don't. And if it's a little bit different, it doesn't matter. Your cells are still going to attack it, but that doesn't seem to be the case. So you can have a better fit than, or there's better fits and not such good ones. But the main thing is, so keep that in mind, and it makes it's an important it's an important feature of each of our cells, because it helps in immunology. It helps the body to recognize non-self and to be able to get rid of microorganisms and non-self cells, because that's what it's supposed to do. They're also known as human leukocyte antigens, but I would say MHC is the one that most people know. And then there's some, and we'll get to them, but there's also some that display the MHC2, and that's usually the white blood cells, the B lymphocytes. So just the B lymphocytes usually have, might be a few others, but maybe the macrophages. But there's another complex that they have in addition. They still have MHC1. So MHC1 is on every single cell, even if they have MHC2 in some cases, and it's only in white blood cells, they still have that one. And that just means your cells, every single cell, and that doesn't matter where it is in your body, except for the red blood cells, has a code on it that says it's yours. And that's important for the immune system to recognize that. We already talked about this, so there's histo and compatibility, so it's accepting, your own body is accepting your own cells, which is good. And like I said, the MHC really stands for the genes that code for it, but you really need, that's just a minor distinction. MHC is basically what they call the receptor. And then, like I said, in addition to the B cells, macrophages and dendritic cells, you can go back in your book. So those are the main macrophages, the phagocytic cells. They have a class 2 on them. And that class 2, which is this one, is important because these cells, so these three cell types, macrophages, dendritic cells, and B cells, specifically the B cells, are involved in presenting those cells, presenting antigens to T cells. So somehow, in addition to having these to recognize the tissue, those specific white blood cells have these receptors that are actually at some point presenting a foreign molecule or a foreign structure to T cells, and that's how T and B cells work together to fight off invading microbes.
So the role of the MHC2 is to grab pieces.
Yeah, and present it. So they're basically connecting to an antigen and then coming to the surface, and then the only way a T cell can actually, then be active and maybe kill that cell, or and it will do that, is by having a connection of, by recognizing not only antigen, but also that MHC2. So it's a signal for that T cell to say, okay, this is something I need to be taking care of. But it's almost like a safety issue that T cells have to be presented with both of those to make sure they're not just destroying a cell. They know they're destroying the right cell because it's presented by a B cell on their MHC2 receptor along with the antigen. So it's a combination of those two. And those cells that are able to present these antigens to T cells are called antigen presenting cells. That's I don't know how they came up with that name. So again, what then the receptors help with is to help the lymphocytes that are floating around in your body to find specific or to recognize certain patterns of microorganisms and then to act against them. And B cell receptors usually, so the receptors of B cells usually bind free antigens. So they're the ones who have receptors that they can actually bind those antigens and then later on present them. And then the T cell ones as I just said, they bind the process antigen by those B cells along with their MHC molecules, the MHC2, that are presented to them. So they bind the combination, not just the antigen, but also the MHC2 molecule. So the way we just went through the cells that present, does anybody recall at least one cell that presents antigen? What's an antigen presenting cell example?
B cell.
B cell. And the question now is, so we said, you know, a pollen can be an antigen, or so that's where more of an allergic reaction comes in, or it could be you know, microorganism. It could be a lot of things. I mean, anything foreign really with a structure, and we're going to look at antigens and what creates a strong reaction and what doesn't. But if you think of how many antigens are coming into our body, how do the B cells have the right receptor for it? So if they, you know, there's no more pattern recognition. They're not looking for that. They have to find a very specific structure. So if you have a pollen from, you know, especially in the Central Valley, there's nothing but pollen flying around all spring, and you've got that coming into your body, and they all look different, and they all have different structures. How does your body, and then you think of all the microbes that we're exposed to, how does your body have B cells with the right receptor for it? Because they don't recognize a bunch of things. And that's one of the things I left out. The answer to this I left out because it's so complicated. But I'll say a few things about it, because I think in your book it's a whole diagram of how that works. So what you have is, in comparison to other genes that are always, so you have your genes and they're red and they produce protein. Remember that, like from our experiment, you know, we had the fluorescence protein red, and so usually you have, you know, a gene red, and there's structures that code for a protein or whatever, usually protein. Then there's structures in between, but usually you translate DNA into a protein. For the lymphocytes, you have 500 genes to produce. So you have 500 genes to produce a huge amount of those, and there's a few genetic mechanisms that you can look up in the book that basically create an unbelievable amount of different B cells with different receptors. And so you have all these, and it's kind of it seems like kind of a waste of energy, but that's how it works. It's producing those ahead of time without knowing what's coming to the body. But then you have, because it produces all these single lymphocytes with a single specific receptor, the chances are that whatever comes into your body finds one B cell is going to be ready to bind to it, or to bind that antigen. And if that C cell binds, it starts producing, like, to multiply like crazy and produce these clones, and then you have a huge amount of B cells ready to work against this one specific antigen. So first, and they call it clonal selection. So first you have you have undifferentiated lymphocytes initially, and then they have a lot of divisions and genetic changes, and you create millions of different cell types. They're all B cells, but they all have different receptors. And then each of them has a unique receptor. And so if you take this and you say, these are all B cells, and they all have, I mean, this is 5 out of billions, and so you have 5 here, and they all have specific receptors, and then you have an antigen that looks like a triangle. This B cell will get to the site and recognize that, bind it, and then that initiates this immune response where this one specific B cell is starting to produce a huge amount of clones. and then that can react to the rest of them because you have a higher concentration. You wouldn't just do it with one B cell. So there is all kinds of mechanisms involved, and say you have different cell area, different genes for say and we get to the receptor in a little bit, but say all these genes code for say for a variable area on your receptor. I don't want to make this complicated, but they can basically, you've got tons of those, and they can basically just use one of them and eliminate the rest. And for the next one, they use this one, and for the next this one, and then you combine it with all these other genes, and because of how they just choose specific, or how these specific DNA or genes are chosen, and the combination of them creates this vast amount of possibilities. And then you have all these B cells, and then the ones that are activated by getting in contact with what they're built for are building clones, and then you can have a decent immune response. So a few things about those B and T cells. I said before, how you remember it probably is the T cell, they're all coming from the bone marrow, and then the B cells stay in the bone, so be in the bone marrow, and then differentiate to a B cell with a specific receptor, and then that migrates to specific lymphoid organs. So the lymph system, and especially the lymph node, we talked about how that's the space, how that's the area where a lot of the microorganisms are being attacked or encounter the immune response, the immune cells, white blood cells. So that's where they end up. But they're differentiated, and they mature in the bone marrow, and then move to the lymph node. And the same thing happens to the T cells, except they end up in the lymph nodes, but they're differentiated, and they mature in the thymus. So that's probably why they call them B and T cells. All lymphocytes have the same receptor specificity. What did I just tell you? No? False, yeah. So they're all different. Very false. So now, to talk about, or to go into a little bit more detail, and I've tried to make that's a really complicated process too, but to make it less complicated, I'm not going to spend too much time and just say the basics. So immunoglobulins are the receptors. So these are the receptors we're talking about on B cells, and this is what I was showing you here with the variable and the constant. So this is just their main structure. They're made out of a heavy chain and a light chain. So the light one is the short one. Light just meaning smaller and with less molecular weight. These are heavy. And so up here is where they bind the antigen. This is where they bind. So this region, this constant region, doesn't really mean too much. It's not that important. You just have this area that attaches to the membrane of the B cells, but this has to vary, because for every single antigen you want a different, or you need a different receptor. So this binding site is where the variability happens. So immunoglobulins is a large glycoprotein, and that's the specific receptor to the antigen. And how the variability, how you get these billions of different B cells that are specific to one antigen, is by changing what the variable site looks like. And this is the same thing. So if you see that structure, it's a Y-shaped structure. Just know it's a immunoglobulin. There's two heavy chains, 2 light chains, they're identical, and you have the antigen binding site that's different for each single one. And this is pretty much what we already talked about. So you have different chromosomes in your body that code for it. Number one, there's a bunch of genes that code for the variable region, and a lot of processes, genetic processes that make this great variety of B cells. And then, so you have this production of all these different B cells, as it shows here. Here. So you have all these produced in the bone marrow, and then once they're synthesized, the immunoglobulin is brought to the cell membrane and then acts as a receptor, and then that's the one. So you have this all these different genes, all these different chromosomes that all together make up what one of the B cell receptors is going to look like, and then when they come to the cell surface, that's the receptor it has. So this one cell has a specific this one specific B cell has a certain receptor, and that receptor stays with it until it's done its work, or until it dies from age. So there's no changes of. It just has this one, and it might find a job because there's an antigen out there that works. It might not. It might just sit around for the duration. And then T cells, and that's just because we're talking about receptors. T cells also have receptors, and they're a little. I mean, they also have processes, genetic processes producing those. So they have also constant and variable regions. They're they're smaller, and they're just they're 2 chains instead of the 2 heavy chains and the 2 light chains. But we'll focus mainly on the antigens. So what happens is, in the bone marrow, remember that's where the B cell maturation happens. In the bone marrow, you have stromal cells. They're like bigger cells that nurture lymphocytes, and so they nurture lymphocytes. They provide signals for them, and then once they develop their specific receptors, they move to the lymph nodes, the spleen, or other areas. We talked about malt and gall, these areas where white blood cells in mucus or in the gastrointestinal tract move to. So all it is these B cells develop in the bone marrow and then move to areas where they can actually get in contact with antigen and start doing their work. And that we already talked about. So they have this immunoglobulin as a surface receptor for antigen. And in the T cell maturation, as I said, that happens in the thymus. And what they have is these receptors that we looked at. That's the only thing you need to know is there's CD4 and CD8. So they're called cluster of differentiation. I have to remember that it's a CD4 receptor and a CD8, and some are helper cells, so you have T helper cells and you have T cytotoxic cells. So those obviously, cytotoxic means those ones are the ones that can actually destroy, and then the helper cells we'll talk about what they do. And so once they are mature they're mature and they get out of the thymus, they move to the lymphoid organs. And just so you get an idea, I mean, there's 25 times 10 to the 9 T cells each day traveling between the blood and the lymph. So you can imagine, when I say, the B cells, there's this vast variety of B cells and their receptors. That's you can imagine if you have that amount of cells, you have cells for almost any antigen possible. So we talked about antigens enough, and clearly there's differences between what that means. You can have a microbe entering your body. You can have something like a pollen enter your body, maybe blood from another person. There's different they have different sizes, shapes, etc. And most important is usually they're recognized as non-self, number one. And if they're too little, they don't really do that much. So it's good if they have a molecular weight of over 10,000. So if you would have a glucose, you have one of the glucose molecules that we use for energy to metabolize. It wouldn't. These are usually smaller. A lot of the proteins or a lot of the more simple macromolecules are smaller than that, so they're not inducing an antigenic response. But there could be, I think in food allergies you probably see a response to specific molecules that are smaller. And so if you look at a specific antigen, then they have a certain structure, and that structure is recognized by the immunoglobulin. So this receptor in the B cell recognizes this structure, and if the structure is complex, it's a little bit easier because they can attach here and they kind of recognize these. Then you have really poor antigens, mainly really small ones, or some that are really simple or repetitive. So that means you can imagine that if you have something simple like that, it's not recognized as easily as a complex microorganism that surface looks like this, and the antigens are able to recognize that immediately. So there's a little bit of a difference. They probably recognize a lot of different structures, but there's a difference between how strong of a response is initiated by the antigen itself. And then what can be done is that if you connect one of those, they call them haptens, but basically they're one of those examples where you have a really small molecule and it doesn't do anything. It's not really recognized by the body. But if you connect that to a carrier molecule, then it's big enough, and it creates it first of all creates a sense of direction for the antibody, and it also for the immunoglobulin, and it also has is more complex, and so then it can create a immunity against it. So it just shows you that these very simple antigens are not always eliciting a response, whereas if you connect them to a carrier, it might do that. So that's important because that's what we do now in therapy, where we try to connect drugs, for example, to attack specific cells, and how they do it is to take you take whatever you want the immune system to eliminate and attach it to a carrier, for example, so it makes it easier for the body to react to it. So if we know this is part of a specific pathogen we want to destroy, we'll use that, add something to it, and then the whole structure together is going to be recognized. So those are all just categories. I'm not going to go too much into those. I already said blood groups are an example of antigens that actually occur in the same members of a species. So in our species, we all have surface markers on blood cells, but they differ. So we have different blood groups. Or we have super antigens, just some that create a huge response that can be overwhelming and dangerous. Allergens or antigens that evoke allergic reactions, pollen, etc. And then some, I talked about that too, in autoimmune disease, where you have your own body recognizing your own tissue as foreign. Maybe we should I'll do a little bit more and then if you get another break. I said it's just if you take a break now, then we have a whole hour. So let's take a break. It sounds like we need one.